All right, this video is going to be all about chromosomal mutations. There are two types of mutations. There are gene mutations, which are things like sickle cell, where you have like an A that gets changed to a T. So very small, tiny mutations that affect one gene. And then you have chromosomal mutations, which affect chunks of genes and therefore, or chunks of chromosomes and therefore multiple genes. And so that's what we're looking at in this, because we're talking about meiosis and chromosomes and karyotypes and those kinds of things. So the first type, there are 5 different types of chromosomal mutations, and the first type is a nondisjunction. All right, junction is where things come together. So like two roads would come together at a stoplight, and that stoplight, that intersection would be the junction. To disjoin, or a disjunction, is where things come apart, where they can be taken apart. And so when something in meiosis, your chromosomes should disjoin. They should separate out. When they don't, it's called non-disjunction because they're not disjoining. And so that's all non-disjunction is. It's when the chromosomes do not separate in either meiosis 1 or meiosis 2. If it happens in meiosis 1, all 4 of the gametes are going to be affected. If it happens in meiosis 2, half of them will still be normal, but the other half will be affected. All right, the results of non-disjunction are going to be monosomy or trisomy, or sometimes even more than that. So you can see in this picture, these cells down here have trisomy, and this one. These are monosomy, they're missing a chromosome. So for humans, this would be 47. This would be 45. Over here, again, 47, 45, but these would be normal and healthy at 46. And so that's the result is extra chromosomes or not enough chromosomes. And it can happen more than once. So it may be that you have 48 chromosomes. You have an extra 13 and an extra 21 or something like that. So it can happen more than once. I can explain how a karyotype is used to diagnose a genetic disorder. Well, it really depends on the genetic disorder. A karyotype is a picture of chromosomes taken during metaphase, so when they're all kind of lining up and easy to find. Chromosomes are matched by size, so that's how and banding patterns as well. That's how they figure out which 2 are number 1, which 2 are number 2, and so on and so forth. Any large chromosomal mutation, like a chunk of a chromosome is missing, or a chunk has been added, or flip-flopped, or an extra chromosome is there, or there's missing a chromosome, that's going to be seen on a karyotype. Now, something like sickle cell, where it's simply a substitution mutation where one letter was changed, you're not going to see that on a chromosome because it's not changing the shape or structure of the chromosome. And so karyotypes can be used to diagnose chromosomal mutations, but not gene mutations. I can describe a deletion mutation. Let's get back to my lovely art here. As a type of chromosomal mutation. So a deletion mutation, now these are not genes. This is not gene A, B, C, D, E. It could be, but in general it's not. It's a section of the chromosome. What you're going to see with a deletion is you're going to see a whole chunk, in this case chunk B, is gone. And so if chunk B is 1 gene, that 1 gene is gone. If chunk B was 14 genes, then those 14 genes are gone. And it may not even be, it may be that this break occurred in the middle of a gene. So you may have cut a gene in half and you've lost its function because it's not going to make a protein now. So anytime you delete sections, you can have some pretty serious consequences. Cru-du-Chat is an example of this. The end of chromosome number 5 is deleted. And there are multiple things that we see with those patients. All right, a duplication, or sometimes you'll hear it called an insertion, is where you have a segment of chromosome that is copied for whatever reason twice, and so it is inserted twice. And so here we have, instead of missing B, now we have 2 Bs. And you're like, oh, what's the big deal? You can have extra genes. Well, you don't always want extra genes. Sometimes you don't need extra proteins floating around. If they're doing a pretty serious job, you don't want double them doing serious job because that can create problems. But another issue is, again, if there was a gene that spanned this break in between B and C, and you just inserted another whole gene into that break, you've broken apart that gene. And if it was an important gene, it can create all kinds of problems. So it's not just that there's extra DNA, which can be a problem in its own, but it's the fact that you've inserted that extra DNA into a place that may have created some havoc. All right, inversion. To invert means to flip-flop. So if this is my pen one way, This is inverted, okay? And so what we're going to see here is we're going to see a section of chromosome got broken off and it got reattached, but when it got reattached backwards. And so this BA part got broken here, and when it got reattached in reverse. That can be bad because it may be read wrong, so it may be read backwards. It can also be bad, again, because if there was a gene right here where this B and C were joined and you just flip-flopped it and put A there, the front of A, no doubt, then you've disrupted that gene, and so that can be a problem. And then the last type of chromosomal mutation is a translocation, and this is going to look a lot like crossing over. The difference is crossing over occurs in homologous chromosomes, so like mom's number 1 and dad's number 1. A translocation is happening in 2 different chromosomes, like number 9 and number 22. But the same kind of concept is going to happen. A piece breaks off and gets reattached. Trans means across a distance, and so these chunks of chromosomes are being relocated across a distance, you could say. And so what this is going to look like, and you may or may not even be able to see this on a karyotype. It depends on if it changes the length of the chromosome. So what we have now is we have L and M got put on chromosome number 9 and A and B got put on chromosome number 22. And so again, it could disrupt genes where they were originally set. It could be that this chromosome is read more frequently or whatever than this one. All kinds of things can happen. And sometimes it's not an even swap. Sometimes you might still have, so another option could be DE. And this one would be easier to see on a karyotype. Whoops, that would be O and P. Sorry. This one you might not see on a karyotype because the structure of the chromosome hasn't really changed, but this one you for sure would, because now this one is longer and this one is shorter. And so Philadelphia chromosome is an example of that, where a piece is broken off and added to the other. It's not an even switch. But even when it is an even switch, doesn't mean there's not going to be problems. Both of these can create significant problems for the person who had that translocation. So those are the 5 chromosomal mutations: nondisjunction, deletion, duplication, inversion, and translocation. I hope that helps, but let me know if you've got questions.